morning students. Today you got to see there is another topic, the viruses. Virus, there is an important and also interesting topic. Here you can see the headlines of newspaper in recent times. Have you heard about the terms Ebola virus, Zika, AIDS, SARS, H1N1, etc. and coronaviruses? There are here the serious entries which are considered as biological puzzles. These are nothing but the biological puzzles and cause disease in man. And these are called viruses. They are smaller than bacteria. We have learnt about the attributes of living world in a previous chapter. We already studied. Now you have to pass on about the viruses in a living and a non-living world. Here, the word virus is mostly derived from Latin meaning a poison. The word virus is derived from Latin from poison. Viruses are submicroscopic, obligate intracellular within the cell, parasites. Obligate intracellular parasites, they have nucleic acid core surrounded by a protein coat. It will present. Nucleic acid is there. It is mostly surrounded by a protein coat that is called capsomere. Viruses in their native state contain only a single type of nucleic acids, which may be either DNA or otherwise RNA. The protein coat is there. Along with this protein coat, there is a nucleic acid is there. That nucleic acid, DNA or RNA is there. The study of viruses is called virology. Then, what are the milestones of virology? What are the milestones of virology is that? Here, Edward Jenner, 1796, used vaccination for smallpox. Adolf Meyer, 1886, demonstrated the infectious nature of tobacco mosaic virus. And also the Dmitry Ivanitsky, his contribution is very more, proved that viruses are smaller than bacteria in 1892. Then 1898, M.W. Beijering defined the infectious agents in tobacco leaves as Contagium vivum fluidum. 1915, F.W. Twart identified the viral infection in bacteria, and Harley coined the term bacteriophage in 1917. In 1984, Luc Montagnier and Robert Gallo discovered the HIV virus, human immunodeficiency viruses. Okay, the next one is size and shape of viruses. Here, viruses are mostly ultra microscopic particles. They are smaller than bacteria and their diameter ranges from 20 to 300 nanometer. That is 1 nanometer is equal to 10 power minus 9 meters. Bacteriophage measures about 10 to 100 nanometer in size. The size of TME, mosaic virus, is 300 into 20 nanometer. You can see the adenoviruses, tobacco mosaic virus, hexagonal shape. Then also there is a bacteriophage, T4 bacteriophage is there. Then generally viruses are of three types based on shape and symmetry. Generally viruses are of three types based on shape and symmetry. Cuboid symmetry, example adenoviruses, herpes viruses. Then helical symmetry, example influenza viruses and the TMB. Helical viruses symmetry, example influenza virus and the TME viruses. Then complex are atypical viruses, bacteriophage and vaccina viruses. These are the structure of that viruses, cuboid, helical and complex. Then characteristic features of viruses, living characters. Characteristic features of viruses in living characters and non-living characters also there. Here, first one is presence of nucleic acids and protein. In a virus, it will present nucleic acid is there, that is DNA or RNA is there, most probably the RNA, and protein codes also there. Capable of mutation, it will also having the mutation capacity. Ability to multiply within the living cells, it also having the multiplication work in the living cells. Able to infect and cause diseases in living beings. It will infect and cause diseases in living beings. So irritability, and also it cause some irritability also there. Host specific also there. These are the living characters also it will present in viruses. And some of the non-living characters also it will present. It can be crystallized. Absence of metabolism also there in a non-living category. Inactive outside the host. Inactive in outside the host cell, do not show functional autonomy. Energy producing enzyme system is absent in this non-living category. Here crystallized manner, W.M. Stanley only crystallized the viruses in a tobacco mosaic viruses. Then classification of viruses. Next one is what are the classification occur in viruses. Here among various classification proposed for viruses, the classification given by David Baltimore in the year 1971. The classification is based on mechanism of RNA production, the nature of the genome, single stranded, that is SS, or double stranded, RNA or DNA. RNA is there or DNA is there. Use of reverse transcriptase RT is there. ssRNA may be plus sense or minus antisense. Viruses are classified into 7 classes. You can see in a table manner. Okay. Viral genome. Each virus possesses only one type of nucleic acid, either DNA or otherwise RNA is there. The nucleic acid may be in a linear or circular form. The nucleic acid may be in a linear in condition or circular in form. Generally, nucleic acid is present as a single unit, but in wound tumor, virus and influenza found in segments also there. Okay. Viruses possessing DNA are called deoxyviruses. Viruses they are having the DNA are called deoxyviruses, whereas these possessing RNA are called riboviruses. Majority of animal and bacterial viruses are DNA viruses. HIV is the animal viruses which possess RNA. Plant viruses mostly generally contain RNA, except the cauliflower mosaic virus possesses DNA. Here, the nucleic acids may be a single stranded or double stranded. Anything is there. On the basis of nature of nucleic acid, viruses are classified into 4 categories. They are viruses with the single stranded, parvoviruses, double stranded bacteriophages, that is ssRNA, TMV, and double stranded RNA wound tumor viruses. We already studied something about the different classes of viruses. You can see class 1 and 2, 3, 4 and 5 and 6, 7. Totally 7 classifications is the 7 classes are there. Viruses with the double stranded DNA, viruses with the plus sense single stranded DNA, viruses with the double stranded RNA, viruses with the plus sense single stranded RNA, viruses with the minus sense single stranded RNA, viruses with the plus sense single stranded RNA, plus and minus, single strand, double stranded RNA, anything is there. And also the last one, RT, reverse transcriptase, that replicate with the DNA intermediate in the life cycle. Viruses with the double stranded and the reverse transcriptase that replicate with the RNA intermediate in life cycles. Hepadoviruses. So you can see some of the examples: adenovirus, parvoviruses, rioviruses, togoviruses, rapidoviruses, retroviruses, and rapidoviruses. Okay, the next one is tobacco mosaic virus, TME mosaic virus. The tobacco mosaic virus was discovered. Tobacco mosaic virus was discovered by 1892 by Dmitry Ivanovsky from tobacco plant. Viruses that infect healthy plants through vectors like aphids, locusts, etc. The first visible symptom of TME is discoloration of leaf color along the veins and show typical yellow and green molting, which is the mosaic symptom. The downward curling and distortion of young apical leaves occurs. Plant becomes stunted and yield in affected structure. There is the next one is structure. Electron microscopic studies have revealed that TME is a rod-shaped helical virus measuring about 300 into 20 nanometer with a molecular weight of 29 into 106 daltons. The virion is made up of 2 constitutes a protein code called capsid and a core called nucleic acids. And also the protein code is mostly made up of approximately 2130 identical protein subunits are called capsomeres. Protein subunits are called capsomeres, which are present around the central single stranded RNA molecule. The genetic information necessary for the formation of complete TME particle is contained in its RNA. Here the RNA consists of 6500 nucleotides are there. Next we pass on to the bacteriophage. Bacteriophage is there. What do you mean by the bacteriophage? There is a virus which will affect the bacteria, that is called bacteriophage. Phage means viruses. Viruses infecting bacteria are called bacteriophages. It means there is a eaters of bacteria, phagian, to eat. Phages are abundant in soil, sewage water, fruits, vegetables and milk. Structure of T4 bacteriophage. Here The T4 page is tadpole shaped and consists of head, collar, tail, base plate and fibers. The head is hexagonal, which consists of about 2000 identical protein subunits are there. The long helical tail consists of an inner tubular core which is connected to the head by a collar region. That is collar. Sorry. Here there is a base plate attached to the end of tail. The base plate contains 6 spikes and tail fibers also there. These fibers are used to attach the page on a cell wall of bacterial host during the replication. The double stranded DNA molecule of about 500 micrometer is tightly packed inside the head. The DNA is about 1000 times longer than the page itself. Okay, the next one is multiplication or life cycle of pages. Multiplication, it will occur in some of the pages also there. That is called life cycle. Here the pages multiply through 2 different types of life cycle. In which way the pages are multiplied? There are 2 categories. First one is lytic or virulent cycle. Another one is lysogenic or avirulent life cycle. Lytic or virulent, lysogenic or avirulent cycle. You can see the cycle of that lytic cycle and lysogenic. First one, there are 5 steps are there in a lytic cycle. First one is absorption, penetration, synthesis, assembly and maturation, then release. Here in a first stage of absorption, it will attach to the bacterial cell. The virus, it will attach to the bacterial cell. Then penetration, the nucleic acids of virus page DNA, it will enter inside these bacterial cells. Then synthesis, then it will synthesize new nucleic acids cells of host cells. Then last one is assembly and maturation. The nucleic acids on the DNA, that is called nucleic acid DNA or RNA, anything is there, and some of the parts, viral parts are mostly synthesized, then assembled and matured. These are joined together. Then lastly it will released. The mature virus are released from that bacteria cell. There are number of bacteria cells are produced in a single bacteria. Then it will number of bacteria release of new page particles in this bacteria cell. That is called a lytic or virulent cycle. In that lysogenic or avirulent type, there is a bacterial cell is there, a page, it will attach to the bacterial cell. Then the nucleic acids enter inside this one. Then the circular page DNA is produced. That form, that circular DNA is attached to the plasmids of that bacterial chromosome. That is called that the bacterial chromosome, it will attach that the particle is nothing but the plasmid is nothing but propage. Then it will divided with the help of the bacterial plasmids. It will divided into 2 cells. Plasmids are again, it will divided into 2 way. Then the way the viral particles are only produced. This is nothing but the lysogenic cycle. Okay, you can see one by one. Lytic cycle. During lytic cycle of page, disintegration of host bacterial cells occurs and the progeny virions are released. The steps involved in a lytic cycles are follows. First one is adsorption. Page T4 particles interact with the cell wall of host E. coli. Then the page tail makes contact with the tube and the tail fibers recognize the specific receptor sites present on bacterial cell surface. The lipopolysaccharides of tail fibers act as a receptor in pages. The process involving the recognition of page to bacteria is called landing. When the contact is established between tail fibers and bacterial cell, tail fibers bend to anchor the pins and base plate to the cell surface. This step is called pinning. Then penetration. The penetration process involves mechanical and enzymatic digestion of the cell wall of the host cell. At the recognition site, page digests certain cell wall structure by viral enzyme lysozyme. After pinning, the tail sheath contract and appears shorter and thicker. After contraction of base plate enlarges, through which DNA is injected into the cell wall without using. There is a metabolic energy. The steps involve injection of DNA particle alone in the bacterial cell is called transfection. The empty protein code leaving outside the cell is known as host. Then synthesis, that is the third step, synthesis. The steps involved in a degradation of bacterial chromosome, a protein synthesis and DNA replication also occur. The page nucleic acid takes over the host biosynthetic machinery. Host DNA gets inactivated and breaks down. The page DNA suppresses the synthesis of bacterial protein and directs the metabolism of cell to synthesize proteins of the page particles only. And simultaneously, replication of page DNA also takes place. The last one is assembly and maturation. These are joined together, then matured. The DNA page and protein code are synthesized separately and are assembled to form page particles. The process of assembling the page particle is known as maturation. After 20 minutes of injection, about 300 new pages are assembled here. Then last one is the release. The page particles gets accumulated inside this host cell and are released by the lysis of host cell wall. These are up to lytic or virulent cycle. Then lysogenic, there is a simple concept. Lysogenic cycle, page DNA gets integrated into the host DNA bacteria cells and gets multiplied along with the nucleic acids of the host. No independent viral particle is formed. As soon as the page injects its linear DNA into the host cell, it becomes circular and integrates into the bacterial chromosome by recombination. The integrated page DNA is now called propage. The activity of the propage gene is repressed by two repressor proteins which are synthesized by page gene. This checks the synthesis of new pages within the host cell. However, each time the bacterial cell divides, the propage multiplies along with the bacterial chromosome. On exposure to UV radiation and chemicals, the excision of page DNA may occur and results in lytic cycle. Then some of the topics are there. What is virion? Virions. Okay, virion is an intact infective virus particles which is non-replicating outside in a host cell. It will not replicate in outside the host cell. Viroid is a circular molecule of single-stranded RNA without a capsid and was discovered by Stewart Einer in the year 1971. The RNA of viroid has low molecular weight. Viroids has citrus, hexacortis and potato spindle tuber disease in plants. And virusoids were discovered by J.W. Randles and co-workers in 1981. They are the small circular RNAs which are similar to viroids, but they are always linked with the larger molecules of the viral RNA. Okay, prions were discovered by Stanley B. Brusner in the year 1982 and are proteinous infectious particles. They are the causative agents for about the dozen fatal degenerative disorders of the central nervous system. For example, Creutzfeldt-Jakob disease, CJD, bovine spongiform encephalopathy, PSE, commonly known as mad cow disease, and scrappy disease of sheep. Okay. Up to this one, you have to read very carefully. Thank you, students. Thank you, Nanda.